Thanks everyone for joining. Today's training is for iGEM teams who are using Benchling or who aren't. If you're new to Benchling, that's great too, and hopefully we'll give you a taste of what Benchling can do for you and your team. So just to get started, I'll give you a little bit of an outline of what we're going to go through today. We'll introduce you to Benchling and briefly to our partnership with iGEM. We'll cover a couple of major challenges today in synthetic biology research. We'll also briefly just give an overview of Benchling's notebook and molecular biology applications, and then we'll dive into a live training of those applications. And at the very end, we'll have about 10 minutes for Q&A. And just to introduce ourselves, I'm Lily Helfrich. I'm on the marketing team at Benchling, and I largely work on our academic program, which includes our partnership with iGEM.
And I'm Melissa Evans. I'm on the customer success team here at Benchling. I work mostly with our enterprise customers, but today I'll be delivering a training for you guys on how to leverage the notebook and mobile applications for a synthetic biology experiment.
Awesome. So briefly, just an overview of Benchling's partnership with iGEM. We have partnered with the iGEM Foundation for this year, and that's largely because we share a lot of similar goals. So iGEM is dedicated to education, competition, and the advancement of synthetic biology, and at Benchling, we too are dedicated to the advancement of biology, and we do that largely by providing scientists cutting-edge tools to carry out cutting-edge research and to collaborate on that research faster. That has been our mission from day one. Just briefly as a snapshot, Benchling was founded in 2012 with the mission to help scientists accelerate their research. Today, we are pushing 130 employees, 20% of whom are in customer success or support roles, which means they work directly with our users, and 30% are in engineering, which means they're working on our product. We have over 200 enterprise customers, so these are biotech and pharma companies that trust Benchling to carry out all of their scientific workflows. And we actually have over 135,000 academics worldwide, from iGEM teams to leading labs at top universities that rely on Benchling to design and analyze their experiments. So briefly, you're all working in synthetic biology, and we know at Benchling that synthetic biology is incredibly complex and poorly supported by existing systems. So synthetic biology, like a lot of large molecule research, involves tracking very complex entities from DNA sequences to proteins, examining rich linkages between those sequences and samples that you generate. You have multi-dimensional data sets, many of which are electronic nowadays, and you have these highly collaborative, super iterative, evolving workflows. And you guys see that on a day-to-day basis working with your iGEM teams. But a lot of current solutions that scientists are still using, like paper and Excel spreadsheets, make it really difficult to capture data, browse and query data, and share that data across your team. And that's why we started Benchling. Benchling is a modern tool built to capture all of this complexity in one place. So Benchling captures biological entities, that's DNA sequences, protein sequences, oligos, captures all of your experimental processes and your results and even some of your analysis. This is all done on an intuitive cloud-based platform, which means that you can access and collaborate on that real-time data from anywhere, and you generate more reliable data. We know that Benchling users record data 85% faster and make fewer errors, and we also know that Benchling users preserve legacy knowledge, so they generate more reproducible data over time. And then this is just a brief overview of what Benchling is, a single platform for experimental design, analysis, and collaboration. And these are the features of Benchling that Alyssa is going to go into in the live training. So Benchling has a notebook for taking notes, standardizing record keeping, sharing protocols. We also have 10 molecular biology tools for sequence design and analysis. That includes an assembly wizard, an alignments tool, sequence annotation, and more. And Finally, all of these tools together allow you to really streamline collaboration, and that's something that Alyssa also will show off in the demo. All right, let's get started with our training.
Hi everyone, this is Alyssa again. I'm going to just take over now and jump right into Benchling. So we're going to navigate out of our slides here and jump into Benchling. So when you first log into Benchling, this is what you'll see. I'm going to start this training off with an introduction to the platform and how you would join an organization or create one. And then we'll jump right into the specifics of the notebook and molecular biology training. So when you log into Benchling, starting from the left-hand side, you'll see this blue sidebar with the jellyfish, and this is your workspace. So it shows you a preview of your recently opened items. I can expand this view to get a calendar view of my experiments, which we'll see in more detail throughout the live training in a second here. I'll just collapse that and continue down the list. After our blue Jeffy P icon, you'll see the projects icon. So this is where all of your entries and all of your biological inventory will live in Benchling. And we'll jump into that in more detail here in a second once we get started on our iGEM 2019 project. Just to point out a few other things, we have a global search button that's accessible here. You can search for any entry or biologic that you have living in Benchling. We have a really robust filtering and search system, so I strongly encourage you to engage in that after our training and once you get going on Benchling. We have a global create button here. From this button, I can create a project, an entry, protocol, or any biologic within Benchling. It's really a master creation button, if you will. Moving right along, I will skip over this button because it actually doesn't impact you guys. This button here is our external data, our external file attacher, so you can connect to your Google Drive or your Dropbox and pull in images and files from there, which I'll also show you guys how to do throughout the training. And lastly, down here at the bottom left-hand side is your avatar, so this is your user icon. If you click this, what you'll see is your picture, of course, for your user settings, and below that immediately are your organizations that you're a part of. So you can see here that I'm part of the Benchling organization, which makes sense, I work here at Benchling. I'm also part of this iGEM Team Terminator organization, which is my faux iGEM team that I will be using for our training today. And if you scroll down here at the bottom, you can join an organization if you click this button. So I'll just show you guys how you can join an existing organization, or in the case that you're just getting started with Benchling, how you'll create a new one. So let's say Lily is my iGEM team leader. She's facilitating the Benchling onboarding for our iGEM team. She has sent me the organization name to join. So that would be Benchling for iGEM. So I can immediately request to join this one here. I can also optionally create a new organization. So if you're the leader of your iGEM team and you want to get started on Benchling, I would encourage you to create an account and then jump here to create now. So I'll just click request to join just so you guys can see how that motion looks in the platform. Great, so hopefully Lily will approve my request, and then I can expect to see that organization here in this listing among these other two. So that's how you would get started by joining an organization or creating one. We're going to jump right into projects next. So projects are the top level of organization within Benchling. So you can create a new project by navigating to the blue plus icon at the top right corner. Just to point out how you would view this queue here, all of your projects that appear in this list are assigned to an organization. So you can see that I can filter based on projects only shared with Benchling or just with my iGEM team organization. And today, since that's what we're working with, I'll just leave that filtered. Just to point out one other thing before I leave it filtered here. If I go to any, I can star any of these projects and they'll live at the top of my projects list. So this project's the most important to me on this account. I'll star it and I'll always be able to access it immediately from here. Just to point out one other note on projects and organizations, all of the project settings for a given project can be accessed by the gear icon once you're within that project. I can click project settings and I can clearly see that my iGEM team Terminator organization owns this project. That's why it was populated in that list for which organizations it's shared with. And I can invite other collaborators and users directly to this project from this modal here. So just to show you guys how to invite the necessary organizations and collaborators to get started. I will point out one other note from the gear icon which you guys might find helpful. You can also export your entire project from here, and you have the option to export as a PDF or an HTML, which can be used to upload to your wiki sites or be attached in any other form as needed. So just wanted to point that out at the top level for you guys before we get started. So now that we're in our iGEM 2019 project, you'll notice that I have two tabs below that, Notebook and Inventory. Notebook is where all of your entries will live, all of your protocols. So this is where you'll be directly making entries and saving them to, is within this tab within this project. Inventory is where all of your biologics live. So any DNA sequences, amino acid sequences, plasmids, genes, you name it, you can upload that here. And what I have here is a basic organization structure that we recommend generally. So you could see I have my genomic inserts, my oligos that correspond to those inserts, cassette assemblies, oligos used for PCR products, specifically primers for sequencing, those sequencing results, etc. I have my Yeast Toolkit here. For my given biological parts, we highly recommend organizing these by part type. So as you can see, I have entry vectors, promoters, coding sequences, terminators, etc., as I go down the line. So this is just to give you an idea of how you would start to get organized with all of your data and how the notebook entries for that data correspond to your biological inventory. I'm going to click this hyperlink to go back to the higher top level folder organization. And I will go back to Notebook to show you guys what that looks like. So you can see the entry we'll be working with here is stored in just this general Notebook tab. I can move it to one of these folders. Since we're going to be going through a cloning workflow today, I would likely move this to the cloning folder. So let's get started on how you would create an entry in Benchling. Again, you would start at the top with the blue plus icon for create. And you can create an entry by hovering over entry, and you could create a blank entry if you're just, running on ad hoc experiment. But what I highly recommend and encourage is creating entries from templates. So these allow you to create entries that are already templatized in such a way that you could easily reproduce and recreate your data and easily compare experiments across multiple days. So we're not going to work with this template today, but I'll just show you how to create one. So you could see that I type in iGEM in my template selection, and I have this iGEM 2019 cloning template with the gene, the method, and the date. So I would just fill these in and click create. And that'll generate a template that looks similar to the one we'll be working with. It's blank, and I can just go through and fill in all of the necessary information. Again, we won't be working with this one, but just wanted to give you guys an idea of how to create templates and some context of why we recommend them and use them. So jumping right into our template we'll work through today. This is our iGEM 2019 cloning template. I have filled in the genes at the top, my assembly method, which will be Golden Gate, and the date. I would just go through and fill this out. So our target organism is, Baker's yeast. Our target pathway is the carotenoid pathway, and just to give you guys a little bit of experimental context for this demo, the idea here is that I tried to clone in this single CRTI gene, which creates the molecule lycopene, into my yeast strain. I was unsuccessful using a previous assembly method. So in this experiment, I will attempt to use Golden Gate cloning to clone this gene in again, yes, and in parallel clone in my CRTY gene, which will convert lycopene to beta carotene. So we'll be creating 2 cassettes using Golden Gate assembly, and hopefully we'll be able to clone those into yeast at the end of the experiment. So I give a summary of that here. I can, since this is coming from a template, I can use these nice checkboxes, which can be added from this top level bar here, where all of your formatting options live. Today we'll be just assembling source DNA, and we'll be assembling cassette plasmids. No part plasmids or multi-gene plasmids in this particular experiment, and we'll be using the Golden Gate strategy. I want to point out one other thing before we continue to move on through the experiment is you can organize your entries by days. So that can be done at the top here by clicking Insert, New Day. And what that will do is give this top level line, as you see here where it says Tuesday. I could switch this. Maybe I started this on the 8th actually, and I could switch that and toggle it back and forth as needed. This day actually corresponds to your calendar view, so any of these checkboxes that are in this entry will actually populate on your calendar, so you could easily keep track of multiple experiments across multiple entries, so long as you're using this insert day here. So now that we've gone through the background and the context of our experiment, we'll just work our way down. So I've attached an image of my assembly strategy. I can preview this in Benchling by clicking Open Preview. So this just gives an idea of how I'm going about this strategy, and it's helpful information to include in the template as well. Moving right along, I've inserted a table here that contains all of my standard parts. That can be done again from this insert icon under table, and then you could continue to format using these various formatting options at the top of the table. We'll jump into more specifics and even highlights of how to use tables in Benchling because there's quite a bit of functionality there. So that's just how to insert. We'll keep moving right along. So in the first part of our template, we go through an experiment design. So before we actually go and proceed to execute the experiment, we can do a lot of the design and modeling in silico within Benchling. So we're going to walk through how to do some of that. First, we're going to start with importing our gene of interest. I've already imported my CRTI gene, so I really just need to import my other gene for CRTY. You can do that by coming here to the top. Well, first we need to navigate to inventory because we're going to be importing a DNA sequence into Benchling. We're not going to import that to the notebook, not just yet. We can go to inventory, click create, and go to import DNA sequences. You have a couple of options. You can drag and drop files that are locally on your desktop. You can search external databases, which is the example we'll do today. You can select a chromosomal region if that's easier, or you can just input a raw sequence, if this is a quick one-off experiment. So today, lucky for us, CRTY is publicly available. You can see Benchling very quickly finds it. You could select what genome, etc. All of these baseline DNA settings here as you come to import this gene. I'm just going to rename it as this clean CRTY name. I like to keep things very clear. And then I'm going to choose what folder to save it to. So since this is a genomic insert, I'm just going to click to save it in my genomic inserts folder so that I can easily keep track of it. And I'll come here at the top and click import. Now what Benchling will do is import it to that folder, and in parallel it will open up that gene in a new tab. So you can see that I have my gene in Benchling now. It's a DNA sequence. I'm just going to move it over here because we're going to get back to that gene in just a second. So I have now imported my gene. Now maybe I want to drag an image of the gene into Benchling like I have here. So you can do that by navigating to the Genomic Inserts folder and simply clicking and dragging the gene and bringing it in. Benchling will give you this nice little render of the gene, and now it's there. I can check that off that I did that. And also, again, if you're already in another spot in this entry, you can reopen that gene by just clicking this open file icon at the top right. So the next part of our experiment really involves planning out our Golden Gate strategy. And what you'll see here, what I've made is a table in Benchling that I can use to map out my various parts for the different cassettes I'm making. So just to go into a little more detail on the strategy here, I'm going to be assembling the CRTI cassette, but in parallel, remember, I'm going to assemble CRTY as well, and I'll be using 2 different assemblies with a different promoter and a different terminator flanking that gene for the other 2 cassettes for the CRTY gene. So I'll be going through the exercise of mapping those out to plasmids I actually have in Benchling and to the insert that I just imported. So I can come here to my second cassette and I can @mention my insert, and I can do that by clicking and starting to just simply type CRTY. Benchling will populate this little drop down here. I could see this is the one I want. It's the nice clean name I gave it. And since I'll be using this in my other assembly, I can simply copy and paste it to insert here. Now remember, I wanted to vary the promoter on this assembly. So here I've already linked it to PREV1. I have PRAD27 here. That is the plasmid I want to use. I'm just going to delete it for the sake of our demo and show you how you can use our split workspace to simplify your workflow. So I'm going to toggle split workspace. And what this does is it splits my current notebook view into 2, and on the left-hand side, it has an attached protocol. You'll notice this looks similar to the table I showed earlier, and that's because it is. So what I can do here is use this to, quickly select which pieces and parts I want in my cassettes and my assemblies. So just for the sake of our demo, I want to use this particular cassette and insert with the PRAD promoter. So I can just come over here, copy the promoter here, click in, and start to @mention that. Since I've already uploaded all of these plasmids into Benchling, it's just finding those and actually actively hyperlinking them there. And notice how this column is now filled in after I linked it to my plasmid. If I uncollapse split workspace and I navigate to the top, you can see here that I'm actually leveraging one of Benchling's more advanced formulas within a table. So I'm going to click Show Formula so you guys can see what that looks like. And you can see I'm actually doing a lookup on this plasmid from my table above to surface what part type that is within my entry. I used the protocol to quickly select which parts I wanted and match them up to my various cassettes. In addition to that, you'll notice that I have this drop-down type for the assembly type. This is what we call a selector in Benchling, and this is another setting that can be toggled within each table by coming to the settings icon and clicking selector. So you can model various drop-downs, again, to repeat and reproduce experiments in a very clean way. I'll just continue to go through. I'll collapse this table since it's quite big so that we can continue to move on through our entry. So now that we've set up for all of our cassettes, I went ahead and already made 2 of our cassettes. So one was CRTI, the other one was CRTY. We're going to go ahead and actually assemble this last cassette using the PRAD promoter with the ADH1 terminator. So now we're going to jump right into Benchling's molecular biology suite. And I can do that by opening up one of my parts. that I will be using in my assembly. So if we go back here, I already have all of these parts open in the top part of my workspace. It's all of these tabs at the very top level. I can click this hyperlink to just navigate and open up one of them, which is how we will get started with our assembly. So I have one of my plasmids open. This will actually act as my backbone plasmid. So to start proceeding with a Golden Gate assembly, what you would do is open up all of your parts in your Benchling. It looks like we're getting some questions. We'll take those at the end. So you would open up all of your parts in Benchling here at the top level and put them in, whatever order you've designed. And then you would click this assembly wizard at the bottom right-hand corner of your screen. This is accessible from anywhere in the molecular biology suite. So basically, anytime you have any sequences open, you can always access the assembly wizard. So we're going to go to create new assembly. We're going to be proceeding with Golden Gate, but as I said earlier, you can also model digest and ligate or Gibson-based assemblies in Benchling as well. So I'll click Start. And what you'll see here at the bottom is my assembly pane. So this is where I can start to select all of my different parts to put together. So I know I'm going to have quite a large cassette here, so I'm going to just go ahead and add in those other inserts. I have a total of 8, so I'll just add in 8 here. I think I'm at 5, 6, 7, and 8. So starting with our leftmost piece, I will come over here to backbone and notice that as I set a fragment, I will really just select these directly from the pane in the molecular biology suite. So I can click right here into the sequence, and I can choose to either set this from a specified selection here. These plasmids I'm working with today are designed to be used in Golden Gate cloning, so they already have BSA1 site recognition sites in them to assemble cassettes. So I'm going to go ahead and just choose BSA1 cut sites. And then I can click Set Fragment. It'll show me a little preview of what my overhangs look like, and it will also confirm that this will be done doing a digest since recognition sites are already present in the plasmid. Moving on through our next couple of inserts, I'll just navigate here to my next piece. This is my promoter, which is PRAD. Same story here. I'm just going to click BSA1 cut sites, click Set Fragment. It shows me that brief preview. Moving right along, now we're to the more interesting piece, in my opinion, is our insert, which does not contain BSA1 cut sites. So this is really the beauty of using the Golden Gate cloning tool, is I will actually, in this piece, I'm going to select my entire sequence here for CRTY, and instead of using BSA1 cut sites, I'll use the selection. Notice underneath it says design a primer pair. So what Benchling's actually going to do is, once I set this fragment, it's going to design and generate primers I can use on this gene for a genomic PCR reaction that will put in those cut sites to actually make this assembly work. So we'll just continue right along. We need to still put in our other pieces, and these are all pretty straightforward. We're just going to be using the BSA1 cut sites for the remainder of the assembly. So you could just click through here. We got a couple more pieces left to go. So really you can put together many parts, many pieces. We're getting to the end here. I decided to pick a large assembly plasmid for you guys to show as a demo, just so that you can see it, get an idea of how you would actually use this tool. So we're at the end here. I'm just going to add my last piece, which is my my Coli1. So we'll just select BSA1 cut sites, set fragment. I'm going to give this assembly a name. It'll be rad CRTY. put together all of our parts in our assembly. Notice I'm prompted to save my sequences and for the assembly and my primers for that insert piece. So the sequence I'm going to save in cassette assemblies under carotenoid CRTY, because this is corresponding to that gene specifically, so I'll click select. And my primers, I want to go under my genomic oligos, and I'll save that under the carotenoid folder. Sorry, I think my internet connection is just a bit delayed here. Apologies about that. There we go. So I've selected carotenoid, and I can just click select to save my primer pair there. Do one last double check, and then click create. So now Benchling is going to look for all those recognition sites. It already checked if the overhangs will match, and it puts it together. So you can see on my plasmid view, it's also color-coded all of the various parts in my assembly. And in addition to that, I have this new sub-tab under my newly assembled sequence called Assembly. This gives the lineage and heritage of what pieces and parts I used, their reverse complements. It also shows my primers that were generated from this assembly that I need to use on my insert gene, and then all of my various design parameters for that PCR reaction. I can copy these primers or export them to a CSV to order them. I'm going to go ahead and click Finalize on my assembly because all looks good. Just wanted to point out one other thing while we're still in the molecular biology suite. I have split workspace toggled right now, and that's why I can see that I have my assembly tab on the right-hand side and my sequence map on the left. So this is very convenient to work, with two views in parallel, and these two views actually talk to each other. So if I go here to plasmid and I select coli1, it'll actually navigate me to that part of the gene, that part of the plasmid here. So these two screens do talk to each other, and I just highly recommend taking advantage of that. It's very useful. Notice one other thing, that the actual places where the BSA1 is going to cut are shown here in this assembly view. So I find that to be helpful just to, get an idea, a physical view of what those overhangs are going to look like. I'm going to go ahead and click Finalize. Notice the note here, I need to finalize to use this in creating alignments or editing the sequence. So this is almost a, pseudo view of what your assembly will be. I'll click Finalize, and now that sequence has been saved and my lineage still remains intact in my assembly tab. So, I'm going to navigate back to our cloning template, and now we can just drag in our new assembly here. The same exact way that we did before. So I'll just take this and place it here. It looks like I misnamed my assembly. That's very easy to change. I'll just come right back to it, come to this Metadata tab here, and you can change the name of any of your sequences by clicking this Edit button and just making that change. There we go. So now I have my two assemblies, both mentioned in my entry, and I am very happy that I'm able to track that. So as we go through here, we won't be assembling multi-gene plasmids in this demo today, but if I wanted to just start to track that setup, I can start to @mention the same way that I did before. So I'm mentioning my new assembly. I can just put that here. I can center it and specify I'll use BSMB1 this time using that same selector. So now I've really set up for modeling my in silico assemblies. I can start to proceed with experimental execution. So how would we track, set up and capture that data in a template in a repeatable way? So that's what we're going to work through next. First part of this would be to set up a PCR reaction for your genomic DNA at the bench. So what I've done here is set up two tables that use, they also leverage all of the formula functionality within Benchling tables, but more simple formulas. So I'll show you these formulas by clicking Show Formulas, and you can just see that I'm using simple math formulas here, but in addition to that, these tables can talk to each other, so I'm also referencing a cell for the number of PCR reactions to calculate my total master mix amounts to get set up for this PCR. So the tables can talk to each other, and you can, you know, cross-reference and use formulas accordingly. So just moving right along, I've set up my master mix for that genomic PCR. I can continue to track my process here, so my PCR tube labels, if I added my primers. So remember I generated primers for that assembly. I can mention those here just to be very clear, and then I even have a spot to measure the final concentrations of these after my PCR reaction. I'm recording my thermocycler conditions below these tables, et cetera, et cetera. The next step would be to set up your PCR reaction for your Golden Gate assemblies. So another table here, different one. So now I wanted to measure all of these concentrations and, you know, calculate how much of everything I need and all of my, you know, according dilution ratios if they're needed. So one other thing to point out, I'm using a lookup formula again from that top level table to reference my plate position for these parts. These parts actually come in a standard plate you can order on iGEM, so it's very useful for me to just have that easily here to surface, so I can very quickly just pull from there, measure the concentration, use it in my reaction. All of these tables can also be downloaded as a CSV, just to point that out. Down here, I've done a little bit of a different setup because I'll be able to leverage a master mix for my other two assemblies since I'm just changing a couple of parts. So just to give you an idea of how you could set that up and, you know, copy and paste essentially from experiment to experiment. Record my thermocycler conditions again and go through the experiment. So I have a couple of other notes here on maybe what you might be doing in the bench top space. So you would proceed to transform these at the bench, pick colonies, mini prep, and prepare sequencing reactions. Maybe you want to, you know, while you're going through your experiment, there may be a couple of other validation steps you would take. So one of those might be to generate some PCR products with some other primers. I won't show you how to design the PCR primers particularly here today, but I have done that for my first assembly. And just to show you that you can generate PCR products in silico and model that reaction in Benchling using the primers you've designed on your particular DNA sequence, and then you can insert your PCR products here. So I included this here just to give you an idea of, you know, how that functionality works and, you know, what you're really going to get out of it. So I've done that here for my initial set. I will not go through that motion for my new cassette. I'm feeling very confident about it. Another thing you may want to do is run a digest. So again, you can model this in silico and Benchling before you go ahead and do it. So I will just open up this assembly here that I had already assembled. I'm going to take off my split workspace to give me more screen real estate, and I'll come here to the plasmid view. So maybe first I want to just get a preview of what cut sites are here. I can navigate to this gear icon and click cut sites and see all of the cut sites. But maybe this isn't helpful enough for me to you know, choose. Maybe I don't know off the top of my head which cut sites I want to use. So I can actually close that and come here to the scissors icon, which is my digest part of the toolbar. So this is where you can start to interact with the digest tool in Benchling. What I'll do here is show you, we're gonna just quickly select, you know, maybe I want to digest right here in my CRT1 gene specifically, and I wanna find, you know, a single cutter for that gene. So as you design digest, you know, you go through a lot of different motions. There's a lot to keep track of depending on what you're trying to screen for and validate. So maybe I just want one enzyme that cuts there. So what I can do is select that region, and down here, I want to show enzymes that cut in the current selection. And I can also filter by the number of cuts in this window, so I only want an enzyme that cuts once within my CR2Y gene. And that's what this is, window here is being filtered for. So I've selected BSA1 in my selection window. I'm going to deselect it by clicking that to get rid of it. I don't want that as part of my digest. So I'm going to just choose this AAT1. It looks like it works well in my buffer 4, so I'll select that. Moving on, maybe I want to also digest in a different region. So maybe now I want to digest in this CAN-R region. So I can, you know, click in current selection. It doesn't look like any of my enzymes only cut once in that selection. So maybe I can click 2. Okay, great. Hindi 3 doesn't have great reactivity in my buffer 4. Maybe I'll just choose this TSPM1 enzyme instead. It gives me 2 cuts there. I'm okay with that. And maybe that's all I want to see for this digest. You can also jump to any of these cut sites in the plasmid based on the enzyme you're hovering over, so this can be helpful. It really all depends on what you're looking to get out of your digest design and your enzyme selection. I'm going to click Run Digest here. And Benchling will populate 2 sub-tabs. So first you get this digest tab where it gives you the enzymes, the cuts, the temperature, and the buffer they are used in. And then it's the fragment information, so what the overhangs are, the cutter, the length of your fragments. I can save this digest, so I'm going to save this as digest 3. Click Save. This digest gets saved to my assembly sequence, so you can see I've performed this with a number of other enzymes and saved it as well. In addition to that, you get this virtual digest tab, which will show you, compared to the latter, what to expect and the relative sizes of your fragments. So again, another way to model experimental process in Benchling and really plan out what you're going to be doing. So I will navigate back. What I often do is I would copy and paste over the table from that digest window right here into my entry just to track that again if I wanted to just see it very quickly. You can also include photos right below of the actual digest results. So you can drag and drop these locally, but what you can also do is leverage the external data connector. So I'm connected to my Google Drive, so I can click there. I can click into gel images and just drag and drop my test digest right over here. It's going to be the same photo. Surprise, surprise. I can collapse this. I can resize this. I can also move these photos around if that's helpful to know. And just have my, experimental results right under my planned. So as we get to the end of the experiment, after you've done these light validation steps, the final step is to design sequencing primers and get this assembly sequenced, right? So what we're going to go through and design some sequencing primers for our assembly. So I'll go back to my assembly. It's already still open here. I'm going to just navigate back to it to jump back into the molecular biology suite. I can click the primer icon here, and you'll notice that I can select attach existing if I believe I have primers that will already attach to my sequence. But for the purposes of today, we're going to create primers. And just to point out one other thing, notice that the 2 primers that came from my initial Golden Gate assembly are attached to this sequence. So I already have 2 primers here. And in addition to that, these primers are considered a pair, so I've linked them together. And just to point out, if you wanted to create a PCR product, you would come here to the Pairs tab after linking two primers together and clicking Create PCR Product. And that would generate a new DNA sequence that would be the amplicon of those primers you've designed. We're not going through that motion today. We are actually just going to create primers using our primer wizard. So you can select your task from here. If I were to do PCR, I would come up here and get a different set of parameters. We're just going to be doing sequencing primers. So I'm going to drag my plasmid map over here, and I'm going to select the region I would like to have sequenced to design my sequencing primers around. So I'm just going to select this part of the plasmid. I'm most interested in to see if the promoter and the insert are in there. So I can click this and click select, use selection. And then I can go ahead and I could specify some other conditions as well. If I have some specific parameters here that I want to define for my GC content and melting point, I can do that. I can also specify the number of results generated. So you may, get an infinite number of primers. Maybe you just want to see, 10. So I can click Generate Primers here. It looks like Benchling was able to find two sets of primers I could use for sequencing. So it gives me the location of these, and it even puts them on the sequence there so you can get an idea of where they are. So you could see that this is the forward primer, this is the reverse primer there. I can select this one, and those also populate on the sequence. There's my forward primer, and then my reverse primer is right there. So you can select both of these, and they show up on your sequence. I also want to save both of these pairs in my sequencing primers folder. So these primers have now been attached to the sequence, specifically the new assembly sequence. And I'm going to also save them in my sequencing primers folder under CRTY. And click select. Click create. And now those are saved in that folder, in addition to being attached to my sequence. So moving on, now that we've designed sequencing primers and at the benchtop we've assembled sequencing reactions using these, the next step, I can also go ahead and drag those in the way I've been showing you guys. I can also mention them here in the entry as well. But let's say you've designed the primers and now you're ready to align your sequencing results in Benchling. So we can go back to the molecular biology suite and go to our alignment tool. So I'm going to go ahead and come back to my CRTI assembly and navigate to this alignment button here on the right-hand side. Click alignments. I've already created one alignment, which I've saved. I want to create a new one. You can choose to align to sequences that already live in Benchling, or you can choose, raw files like AB1 files to align to. So for today, I'm going to use 3 AB1 files generated for my sequencing results, specify my assembly sequence as my template. I can specify my algorithm used. I can even adjust some other advanced parameters there and click Create Alignment. So Benchling will open up the alignment tool, and which you'll see here in a second, and I'll show you guys how to navigate through that, and there it is. So as you can see at the top, we have our template sequence, which is our assembly with annotations, and below are my 3 alignments. Special note is the bottom half portion of this is how you would navigate through your alignment. So all of the parts in red are where your mutations or mismatches are. So you can just see, this is missing from my 3rd sequence. I can come over here and see the votes. I can also see the read number if I hover above here. We can continue to navigate through the sequence. You can also adjust this viewing window to make it less or more detailed. You can, see even more if you stretch it out that far. You can also get very granular if you'd like and make it very small. You can, in addition to that, there are a lot of viewing settings that make this a very nice experience. So you can come up here to this gear icon similar to the other tools in the molecular biology suite and toggle on what you'd like to see. So maybe I want to see where my primers were. I can click primers and boom, you can see there's one of my primers that's attached to my sequence. So you could really make this as custom as you'd like it to be. And that is just a quick overview of how you'd use our alignment tool. I'm going to quickly jump back. We are almost done with our demo, and then we'll get to Q&A here. The last part of the demo is to use our Benchling Sync app. So this is included for iGEM teams to use, and it allows you to check out. You can attach files, such as an Excel file, for example, to Benchling, and you can attach it to your entry and reopen it within the local native application, make edits, save that file, and then return that file back to Benchling. So I'm going to just show you guys very quickly how to do that. Excuses for opening a new tab. I'm logged into 2 accounts for this particular entry, so I need to come over to this account since this is the account that's signed into my Sync app. Don't worry about this, it would not come up for you guys, but let's proceed on with checking this out. So I've attached my fermentation model. Maybe I've scaled this up a little bit and I'm interested to see the results of what it looks like when I use other size fermenters. I can notice this floppy disk icon. I can click checkout. So I can go ahead and click that, and Benchling will, give me this notification about editing the file in the local native application. I'm good with that. I want to edit the file. I want to copy and paste new results into this existing model that's attached to my template. That's just to provide you some context of how you might use this in a template. Maybe I copy and paste over all of my production data here. Let's just, for the sake of the demo, we'll just make a quick change and we'll add, today's date, for example. I think it's the 17th. I will click save. So I've saved this. I'll close this. Notice it says syncing. One other note here is when you've checked out the file, the floppy disk arrow is facing up to check in, and then you also have this edit file icon. I'm ready to check my file back in, so I'll click that, click check in, and then my file was saved and attached to my lab notebook entry. So that is the last part of the demo today. We will move on now to the Q&A.
And just one reminder for all of you before we answer a few questions quickly. For those of you who are new to Benchling, you can sign up on our partners page and you'll automatically be granted extra space and Benchling Sync. For those of you who use Benchling, you know how to sign in. If you have questions or if you'd like access to extra space in Benchling Sync, you can contact support@benchling.com. And those tickets will usually get routed over to me, Lily, and I'll help you get that squared away. All right, awesome. Let's really quickly answer a few questions. So I saw a couple in here about, are you integrated with iGEM? Can you import data from NCBI? The answer to both of those is yes, you can import sequences directly from iGEM or NCBI into Benchling. It's really easy. You can do it through the import DNA sequence button that Alyssa showed off in the demo, and there are lots of different ways to import DNA sequences, but using genes from NCBI or plasmids from iGEM is one of the quickest and easiest ways to get your DNA sequences in Benchling. Another question was, could you sort days within your notebook entry and have the most recent one show up at the top of the page instead of at the bottom? The answer to that one is no, but what we do find with academics who use Benchling is that often people are using one notebook entry for all of their data. And one thing that Alyssa showed off in this demo that I think is really valuable is you should use smaller contained notebook entries like templates. And doing so will mean that you just have a few days worth of experiments in a single notebook entry, and then you can put lots of related notebook entries within a project or a folder to keep them organized. And that sort of contains the amount of data and notes in a given notebook entry, so it's not hard to navigate. One other tip is that if you do use date inserts, you can turn on your table of contents and that'll populate in the top right corner of your notebook entry, and that gives you an easy way to jump from date to date. Alyssa, how do you create a template from scratch?
Hey, I will go ahead and actually show this one on the screen to give you guys an idea. So to create a notebook template from scratch, you can go here to your avatar. You can click over the feature settings button. This is filled up with a lot of experimental templates from Benchling, but you can create new collection and then you could host a number of templates within that collection. So to give you our demo today at the iGEM 2019 collection, and then this is the template for my notebook entry. I can just add a new template by clicking new template there on the left-hand side and associate it with a given collection.
Awesome. Someone also asked if we can attach Fred scores to your sequences. So yes, if you upload trace files. Alyssa showed that off in the alignment tool demo. So if you do upload trace files, you'll be able to see quality scores associated with sequences. And one other question I'm seeing here, is there a list of all of the functions? Benchling has a lot to offer. That's something hopefully we've shown today. For most users, the best place to get started is our Help Center. So help.benchling.com, it's a comprehensive knowledge base and can walk you through most of the features that you are curious about, and will give you kind of an overview of what those look like. Someone else was asking if this will be recorded and shared. The answer is yes, we will send it out to everyone who registered for the webinar, and you can share those with that link with your teammates, and we'll also get this up on our iGEM partner page so other folks can access it there. All right, we're just about out of time. Thank you all for tuning in, and feel free to reach out to Benchling if you have any questions. Thanks so much.
Thanks, everybody. Have fun out there.